Okay, so welcome to this video. In this video, what we're going to talk about is the PI3 kinase AKT mTOR pathway and cancer. Okay, so we want to look at the involvement of this pathway in cancer cells. Okay, right. So what we're going to do is we're going to start off by talking about the basic carcinogenic process. Okay, so what happens in cancer development. Okay, then what we're going to do is look at the PI3 kinase AKT mTOR pathway and have a look at how important this pathway is in cellular growth and cellular proliferation, and we'll understand then why it's one of the key pathways that ends up having its components mutated in cancer. Okay, so there are many components of this pathway that are tumor suppressor genes and many that are proto-oncogenes, and those can end up with mutations in cancer. Okay, so we'll discuss which components of the pathway are the most commonly found mutated. Okay, then what we'll discuss is targeting this pathway, inactivating this pathway for cancer therapy. Okay, now it's fair to say that with one exception, which is a drug used to treat chronic lymphocytic leukemia, okay, called idelalisib. With that one exception, the initial testing of drugs that block this pathway for the treatment of cancer has been quite sobering, okay? It's had only very modest success, and we're going to discuss potential reasons as to why that is. And we think the key reason as to why targeting this pathway hasn't been that effective in preventing the progression of cancer is that we aren't able to give these drugs in high enough doses. Okay, and the reason is that this pathway is actually incredibly important in non-cancerous cells as well. So blocking it can have loads of on-target side effects. So remember, drug side effects can be split up into on-target side effects and off-target side effects. On-target side effects are the side effects you get because because the drug actually binds to the target that we intended it to bind to, and that causes loads of side effects because of how important that target is in the body. Okay, off-target side effects refers to the fact that the drug might bind to loads of things that we never even planned for it to bind to, and that's why it might cause loads of side effects as well. But we think in the case of these drugs that target the PI3 kinase AKT mTOR pathway, forget the off-target side effects. We think the on-target side effects are so large that you can't give these drugs in high enough doses to actually have a decent stab at the cancer, basically. Okay, and we'll discuss potential ways that we might be able to get around that in the future. Okay. Right, so let's start off with the basic cancer development pathway. Okay, so we want to discuss the process of carcinogenesis. How does cancer emerge? Okay, so we're going to discuss the leading theory of what happens in the development of cancer, which is that cancer occurs as a microevolutionary process, okay, whereby cells gradually accumulate mutations, and these mutations gradually build up a more and more aggressive phenotype until you finally get to the level where you have cells that are cancerous. Okay, right. So in this theory, which is the leading theory of how cancer develops, you start off with a normal somatic cell. Okay, so let's say here it is, and it could be a cell in all sorts of different tissues. So it could be a cell of the epithelial of the lungs, a cell of the epithelial of the pancreas, you name it. Okay, it can be any different type of cell. Okay, and basically this cell is going to get a mutation that is going to allow it to overproliferate. Okay, now there are two major classes of proteins that you can get mutations in that will then lead to a cell overproliferating. So let's introduce this concept now. Okay, so the two major classes of proteins are the proteins that come from proto-oncogenes. Okay, and these proteins are involved in promoting cellular growth and cellular division. Okay, The other types of proteins that are involved in controlling cellular growth and cellular proliferation are what are known as tumor suppressor genes. Okay, and the proteins that are coded for by tumor suppressor genes, these are anti-growth and anti-proliferation. Okay, so normally, in a normal cell that isn't dividing, you've got this balance between the proto-oncogenes and the tumor suppressor genes. Okay, and no one's winning, basically. The proto-oncogenes are all saying divide, 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 grow, 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 and then divide. The tumor suppressor genes are then saying, no, don't, stop doing that. Okay, and they're in balance normally. So if you want to tip this balance and cause division, there are two different ways that you can do it. Either you can up the activity of the proto-oncogene gene products, or you can down-regulate the activity of the tumor suppressor gene products. Okay, so this means that this first mutation that is going to occur in cancer, which is that a mutation, it's generally thought to be a mutation that causes overdivision, okay, it can either be a gain-of-function mutation, which I'll abbreviate down to GOF, gain-of-function mutation in a proto-oncogene that's going to cause that proto-oncogene's activity to now go up hugely. So for instance, examples of gain-of-function mutations would be if you amplified the number of genes that you have for this proto-oncogene. So usually you would have 2 copies of most proto-oncogenes, 1 on each of the homologous chromosomes. If somehow you end up totally screwing up and copying a whole new one into one of your chromosomes, then you have 3 copies of the proto-oncogene, and now that's going to hugely increase the amount of the gene product, the protein for that proto-oncogene that you produce, and therefore hugely increase its activity. Okay, that would be an example of a gain-of-function mutation in the proto-oncogene. Alternatively, you could have a mutation that actually affects the structure of the protein itself and results in potentially it being permanently active, whereas previously it might have been inhibited by something, or just increases its activity in some other way, maybe. Okay, so those are all examples of gain-of-function mutations in the proto-oncogene. That would tip the balance because now the tumor suppressor genes that you've got wouldn't be adequate enough to suppress the proto-oncogenes anymore. Okay. Alternatively, you could have a fall in the activity of the tumor suppressor genes. Okay, so you'd need a loss-of-function mutation in a tumor suppressor gene, basically. And often you would need loss-of-function mutations in both of the tumor suppressor genes that you have, because again, you'll have 2 copies of each tumor suppressor gene, generally 1 on each of the homologous chromosomes. Okay, and generally you'll need loss of function in both of those genes in order for the function of the tumor suppressor protein to actually be lost completely. Okay, right. So examples of loss of function mutations would of course be deletions of the gene. If you've completely lost the 2 genes, then that will completely remove the function of that protein within the cell. Okay, alternatively, you could have changes in the structures of the protein, which then lead them to misfold and be totally useless, basically. Okay, right, so those are the 2 main types of genes controlling cellular division. Okay, so this normal cell here is now going to get a somatic mutation, okay, and this somatic mutation is going to just so happen to either be a gain of function in a proto-oncogene or a loss of function in a tumor suppressor gene. Okay, now what's going to happen? Okay, I'll now colour this cell in a different colour because now it's got this mutation. Okay, so let's colour this in purple now. Now what's going to happen is it's going to divide and divide and divide because it now has this over-proliferation property, and therefore what's it going to end up producing? It's going to end up producing a whole population of cells that all have this mutation that means that they over-proliferate, basically. Okay, so you're now going to get a new mass of cells which have all got this mutation that means that they over-proliferate. Okay, now if this first mutation was powerful enough that it causes a powerful enough rise in proliferation that you get a visible lump, okay, then we would call that a benign tumour at this stage. Okay, so it's not cancer yet, it's just a benign tumour. Okay, right, so it's a new mass of cells, basically, that wasn't there previously. Okay, right, so now what what's going to happen is you're going to progress onwards. So that's the first part of the progression towards cancer. Now what you have to do is to actually, for these cells to actually become cancerous cells, they have to acquire a lot of other very dangerous phenotypes. Okay, so actually these cells need a huge number of other mutations in different genes in order to actually get the phenotypes of cancerous cells. Okay, so now what's believed to happen is, in this population of cells that have all got this first mutation that means that they overproliferate, you're now going to wait for one of them to get a mutation in another gene that's going to give them another one of the phenotypes of cancerous cells. Okay, so in this population, these cells will be undergoing mutations occasionally. Okay, many of those mutations will of course be in things that don't progress them on towards cancerous phenotypes. Some of them might even, be in essential proteins, such as the proteins of complex 4, maybe in the respiratory chain, and therefore the cell will die when it gets that mutation. Okay, but eventually, if you wait long enough, one of them will just so happen to get a mutation in a gene that now gives it another one of the phenotypes of a cancerous cell, and therefore takes it towards being a cancerous cell. Okay, so let's say that one of the cells in this population here, let's say this one here, now gets another mutation. So I'm going to colour it in a different colour now, in blue here. And this cell's mutation is now going to take it towards cancer. Okay, so it's going to give it another one of these phenotypes that takes you closer to being a cancerous cell. Okay, so potentially it could just be another gain of function mutation in the proto-oncogene, or another loss of function mutation in the tumor suppressor gene that now causes this cell to divide even more rapidly than the purple cells, and therefore what's going to happen is this tumour is going to firstly get bigger, and it's also going to become dominated now by the blue cells, okay, the blue clone of cells rather than the purple clone of cells. So you will still have some of the purple clone of cells, but now the main cell type that's going to dominate the tumour is now the blue clone of cells. And this is why people describe the carcinogenesis process as a evolutionary process, as a Darwinian-like process, okay, because the cell population within the tumour is evolving, basically. It's going away from being the purple cells to now being this blue clone of cells, being the dominant clone of cells in the tumour population. Okay, so here now are the blue cells, and now we've got very few purple cells left over. Okay, so you can see that the dominant clone within the tumour is evolving, basically, which is why it's compared to a Darwinian-like evolution process. Okay, and then this will continue. In this blue clone of cells now, you will just have to wait for one of these cells now to get a further mutation that gives us another one of the phenotypes of cancer and takes it closer to being a cancerous cell. And this process continues, this evolutionary process continues, whereby the dominant clone of cells in the tumour is going to gradually evolve, basically, okay? And as it does so, the clone of cells is going to become closer and closer to having the phenotypes of cancerous cells. Okay, so let me just outline some of these phenotypes that you're going to gradually build up as you progress towards cancer. Okay, so the key phenotype that everyone can name as one of the key phenotypes of cancerous cells is that they have overproliferative potential. Okay, so they overproliferate. So you're going to gradually build up more and more gain of function mutations in proto-oncogenes and loss of function mutations in tumor suppressor genes, and that's going to lead to you overproliferating hugely. Okay, another phenotype of cancerous cells is genetic instability, and this one's very, very important. Okay, so what do I mean by genetic instability? Genetic instability means that cancer cells undergo mutations far more rapidly than normal cells. Okay, so why is this? Well, cells are getting damage to their DNA all the time that could lead to mutations occurring. Okay, however, why does it not lead to mutations occurring usually in a cell? I mean, it doesn't always work, but usually they don't result in actual permanent changes to the DNA sequence. And the reason is that you have all sorts of mechanisms for repairing the DNA when it's been damaged. Okay, so you have DNA repair mechanisms. Okay, so basically, the cancerous cells, the way that they have this property of genetic instability, this tendency to acquire mutations far more rapidly than normal cells, it's because they have had mutations which have resulted in loss of function in the DNA repair mechanisms. Okay, and if the DNA repair mechanisms go down, then your frequency of acquiring mutations goes up hugely. Okay, so at one of these stages in the clonal evolution of the tumour, one of these cells, so let's say, for instance, in this next step, we're going to acquire genetic instability. Okay, so we've got our blue clone of cells here. One of these cells in this blue clone might now get a mutation in the mechanisms for DNA repair, one of the pathways for DNA repair, which inactivates that pathway. Okay, it will now over-proliferate because remember it's got all of the mutations of the purple and the blue cells. Okay, well in fact it's got all the mutations of the blue cells, which means that it over-proliferates. Okay, so it will produce a whole clone of cells that all have this genetic instability now and over-proliferate. Okay, and now in that new clone of cells, let me show this new clone of cells. So I'll put another clone of cells here. Okay, in this clone of cells, the process towards cancer will now be hugely accelerated because you will get mutations occurring far more rapidly in this new clone of turquoise cells. Okay, so the next process, the next step towards another clonal evolution, the next step in this process will occur much quicker than these processes occurred. Okay, so you had to wait ages for one of these mutations to occur. You had to wait ages for this mutation to occur. Now it's going to accelerate the process. Okay, accelerate this accumulation of mutations because this clone of cells now that we're going to use to generate the cancer now has got this genetic instability and therefore is going to acquire mutations more rapidly. Okay, so that's why genetic instability is seen as a general phenotype of all cancerous cells, because for the actual cancer development to occur in a reasonable timescale, in the lifetime of a human being, it's thought that you need this accelerating step, this genetic instability, which accelerates the process after it's occurred. Okay, right, so that's one of the key phenotypes of cancerous cells. You also need to lose senescence, okay? So let me explain what I mean by this. Okay, so lost senescence is one of the key phenotypes of cancerous cells. So normally, cells that can divide in the human body, so for instance, fibroblasts can divide in the human body, hepatocytes can divide in the human body, smooth muscle cells can divide in the human body. They cannot divide forever, okay? If you take them out of the human body, put them into a petri dish, and let them, give them loads of nutrients, give them loads of space, they will not divide forever. They will eventually stop. Okay, they will get to the point where they can't stop anymore because they have what's known as a Hayflick limit. Okay, there is a Hayflick limit as to how many times the cell can divide. And this is because every time one of these cells divides, so let me show it here. Here's this cell here. Every time the cell divides, the two daughter cells that it produces have shorter telomeres. Okay, so the telomeres are the portions on the ends of the chromosomes. Okay, and the telomeres gradually get shorter every time it divides. So the two daughter cells here will now have shorter telomeres, okay, than the mother cell. Okay, and then these can divide again to produce 4 cells and then 8 cells. But every time, what will be happening is the progressive generations will have shorter and shorter telomeres, and eventually you'll get to a generation where they simply cannot divide again, because if they did, their telomeres have gone basically, and they'd start to cut away their actual chromosomes with the coding DNA, and then you get to the stage where you can't divide anymore, and you've gone into the stage which is known as senescence. Okay, so This was a somatic cell, so it will have had a Hayflick limit. It somehow needs to get around this Hayflick limit, and it needs to not reach senescence, basically. Okay, and the usual way that cells do this is by activating telomerase expression, which extends the telomeres back up again and effectively means that the cells can continue dividing as many times as they want because they can just re-extend their telomeres back up. So that's another one of the key properties of cancer cells. Okay, and then that will be one of these mutations that progresses you onwards, progresses you to a new clone that's closer to being cancerous then. Okay, and finally, lost susceptibility to apoptosis. So I'll call this invulnerability to apoptosis. Okay, so again, cells in the human body which can divide, and there are cells in the human body which can't divide. So for instance, skeletal and cardiomyocytes, they can't divide. Neurons in the brain can't divide. Okay, but the cells in the body which can divide, such as smooth muscle cells, fibroblasts, hepatocytes, these are controlled basically. Okay. If you've got a tissue that is overcrowded, okay, because too much cellular division has occurred, these cells will commit apoptosis. Okay, so let me just show this. So let's say we've got a bit of the liver here where it's now overcrowded because too many cells are divided. What will now occur is some cells in this overcrowded tissue will now commit apoptosis, okay, to reduce the population and make more space, basically. Okay, now, tumours are incredibly overcrowded, okay, so why do they not commit apoptosis? Well, at some point in this progression, you must have got some mutation that made you invulnerable to apoptosis so that you wouldn't activate the apoptosis process and you wouldn't depopulate the tumour population. Okay, so again, that's a phenotype of cancerous cells, and that will have occurred at some point along this progression towards cancer. Okay, right. Now, what I want to talk about is the final mutation that is believed to occur that takes you from being what is a late-stage benign tumour to being cancer. Okay, so you gradually go through this clonal evolution process where the clone is gradually getting closer and closer to cancer. It's acquiring more and more of these phenotypes. Let's say it's now acquired all of these phenotypes. Okay, what is the final mutation that occurs that tips it from being a benign tumour to being a malignant tumour or being cancer, basically? Well, the final mutation that occurs is that it gains the ability to be motile. Okay, so the final mutation is motility, and this is what takes you from being a benign tumour to being a malignant tumour. This is what tips you into cancerous behaviour. Okay, so what now needs to occur then is the final mutation that's going to occur is something that allows the cells to gain motility. So cells are not usually motile. Cells usually stay in their position in the body and do not move around in the body, basically. Okay? But the cells in this malignant tumour have acquired a mutation that allows them to now move around in the body, basically. And this gives them two key properties. It allows them to invade into the surrounding tissue and set up tumours, little extensions of the tumour in the surrounding tissue. And it also allows them to invade into the blood and then set up secondary tumours at other sites distal from the site of the primary tumour. Okay, so let me just describe this. So, previously then, prior to us gaining this final mutation, we had this great big tumour here, but it was contained, okay? It was still a benign tumour, okay? It was maybe pushing on the surrounding tissues as it expands, but it was not invading into the healthy cells, okay? The cells here were static, okay? Now what's going to happen is one cell in this final penultimate clone prior to malignancy is going to get a mutation that's going to allow it to gain motility. Okay, right. Now, generally, cancers are formed from epithelial cells. Okay, so for instance, skin cancer, that's formed from an epithelial cell. Okay, cancers of the gastrointestinal tract, those are formed from epithelial cells. Lung cancer, those are formed from epithelial cells. Pancreatic cancer, those are formed from epithelial cells. Liver cancer, those are formed from epithelial cells. Renal cell cancer, those are formed from epithelial cells. So generally, the major forms of cancer are formed from epithelial cells rather than non-epithelial cells. Okay, so what actually happens usually for these epithelial cells to gain the ability to move is a process called epithelial mesenchymal transition. Okay, and this is a process that was used in the embryo in development, basically. So in the development, if you want to move epithelial cells around the embryo, what has to firstly happen is they have to change their phenotype to become mesenchymal cells. The mesenchymal cells can then move around, and then when they've got to their final location, they can then return back to being epithelial cells. Okay, so if I show this rather crudely here, here's our epithelial cell, which is not motile, it can't move around. Okay, and we're assuming that our benign tumour at the moment is constructed out of epithelial cells which are not motile. Okay, now, in embryology and development, if an epithelial cell wants to move, what it turns into is what's known as a mesenchymal cell, okay, which looks quite different, a very different phenotype. It was supposed to look more different than that. It was supposed to be a sort of spindle shape, okay? And this process of turning from an epithelial cell into a mesenchymal cell is known as EMT. So I'm going to colour in the epithelial and the mesenchymal cells in different colours to highlight how different they are. Okay, so here's an epithelial cell in blue. Whoops. Here is my mesenchymal cell in red. Okay, so I'll just label this up. This is a mesenchymal cell. Okay, right. So the process of EMT stands for epithelial, okay, mesenchymal transition. Okay, and it just means an epithelial cell changing into a mesenchymal cell. Okay, now mesenchymal cells are motile. These can move around. Okay, so now this can move around in the embryo. It can get to a new location within the embryo, and then what's going to happen is it's going to turn back into an epithelial cell by a process of mesenchymal epithelial transition, MET. Okay. Now, this is not usually active in the adult human, okay, it's something that's reserved for development. However, what we think happens is this, in this final transition, where some cell acquires a mutation that then allows it to become motile, what we think is that somehow, and this isn't understood at all really at the moment, somehow this cell is going to acquire the ability to return this process on. It's going to return epithelial mesenchymal transition on. Okay, and what that now means is that this cell here, firstly, it can over-proliferate because now it's got all of those mutations that mean that it can over-proliferate. So it will make a whole population of cells that are all capable of doing this. And what's now going to happen is some of these cells, some of these yellow cells here, are now going to go through the process of epithelial mesenchymal transition. They're going to become mesenchymal cells, and then they're capable of invading into the surrounding tissue. They're capable of moving. So now here's our mesenchymal cell for a moment. Okay, which we'll have in red here. It's on the move. Okay, it can go to some new location, maybe in the neighbouring tissue, and then it can decide, right, I'll return back to an epithelial cell, so it will go through the mesenchymal epithelial transition. Okay, and now it's at this distant site, basically, and now, once it's gone back to the epithelial cell type, it can now over-proliferate. So it will now set up its own little tumour at this site further away from the benign tumour, basically. Okay, so you can now set up multiple little tumours around the major tumour here. Okay, so that's the property of it being invasive. It's going to now invade into the surrounding tissue and destroy the surrounding tissue. Okay, in addition, it gets worse than that. Okay, because what can happen is some of these mesenchymal cells can end up getting into the blood. okay, and then they can get out of the blood at really distant sites. Notably, the most important one here is that it can end up in the brain, okay, so it can go into the brain, and then of course it will go through mesenchymal epithelial transition, return back to being an epithelial cell, and then it will form a secondary tumour in peripheral tissues, basically. And that process of setting up secondary tumours very far away from the original primary tumour here, okay, so this is the primary tumour, that process is known as metastasis. Okay, and it's generally the metastasis to the brain that kills you, okay, in cancer. That's what kills most cancer patients, the metastases that go to the brain, not the primary tumour, it's the metastases that go to the brain that kills the patient. Okay, right. So that's an overview of the carcinogenic process. So what we have seen here is the utter importance of the overproliferation of cancer cells, okay? That's the utterly key phenotype of cancerous cells. In fact, it's believed to be the first driver, the first mutation that occurs to start this whole process off, is that the cell gains the ability to overproliferate and produces a whole population of cells that are all capable of overproliferating, and then any further mutations that you stack on top of that are now going to get amplified because you've already got the overproliferation mutation, and therefore you're going to produce a whole population of cells with this new mutation that you've acquired. Okay, so that's the absolutely key one to set this entire process off. And indeed, generally what will happen is, in this clonal evolution process, you won't just acquire one mutation that causes overproliferation, you'll be acquiring multiple of them. Okay, right, so the PI3 kinase AKT mTOR pathway then, it is extremely important in cellular proliferation, okay? So let me just explain this. We're going to give a very basic overview of what the PI3 kinase AKT mTOR pathway does, and then we'll go into it in detail. Okay, right. So basically, if a cell wants to go from being one cell here to being two cells, okay, firstly, what it has to do is it has to grow. Okay, so the first process in proliferation is firstly for the cell to grow. So growth is the first thing that needs to happen. And then what can happen is this larger cell, this cell that has grown, can then divide into two cells. It can split. Okay? Now, in order for a cell to initiate this process, you need major epigenetic changes. Okay, so what do I mean by that? Epigenetics is all about what controls which genes are actually expressed. Okay, so you know, every cell in your body roughly has the same genome. Okay, it has the same collection of genes in its nucleus. There are a few exceptions, of course. Red blood cells don't have a nucleus. Lymphocytes have that silly old somatic recombination to create their own T and B cell receptors, which means that they end up with slightly different genomes. Okay, but the hard and fast rule that generally applies for all cells is that they have the same genome. They all have all the same genes. Okay, however, epigenetics is different between different cell types. Okay, they all express different collections of genes. Okay, now if you want a cell to start this proliferation process, start this growth process, and then proliferate in two, you have to get this cell to hugely change which genes it's going to express. Okay, so you need to start firstly producing loads of new proteins, okay, so that you've got enough proteins for both cells. So for instance, all of the essential enzymes of respiration are going to have to be copied. All of the cytoskeletal proteins, you're going to have to make more of those because you're going to become a bigger cell. You're going to need more cytoskeletal proteins, okay? And other proteins as well, so such as the proteins involved in the ribosomes, the proteins involved in DNA repair. You're going to have to make more of these essential proteins so that you've got enough for two cells rather than just one. Okay. Also, you're going to have to start expressing all of the proteins that are actually involved in coordinating the division process itself. Okay. So there's going to have to be a huge epigenetic shift, basically, to make a cell divide. Okay, now, epigenetics involves several lines of control within the central dogma of biology. Okay, so the first line of control is that you need to change which genes are actually being transcribed. Okay, so we can call that transcriptional control. Okay, so you need to change which genes are going to be transcribed to cause this epigenetic shift. Okay, the PI3 kinase AKT mTOR pathway is not going to change transcriptional control. Okay, there are other pathways that you need to activate in order to change transcriptional control. Notably, the RAS-RAF-MEK-ERK pathway is a major one which can change which genes are being transcribed. RAS-RAF-MEK-ERK. Okay, and we're not going to discuss that pathway. We just need to note that there are these other pathways which need to occur alongside the PI3 kinase AKT mTOR pathway in order for a cell to divide. Okay, so it's important to note that this pathway cannot cause the cell to divide on its own. You need these pathways as well, okay, that cause the transcriptional changes. Okay, right. Now, epigenetics, however, involves other changes just than transcriptional control. There are other means by which you can control which genes are actually going to produce proteins and therefore be expressed. Okay, for instance, there is also translational control. You can control which mRNAs are actually going to be translated into protein. Okay, so the transcriptional control has now said, okay, right, transcribe all of these genes that are involved in making the cell divide. Okay, the RAS-RAF-MEK-ERK pathway does that. It changes which genes are transcribed and gets you ready for the actual division process. So it's made all of the mRNA now that can make the cell divide, let's say. Okay, however, there's no point making all of that mRNA if you're not going to then translate it. Okay, so translational control also has a major role in controlling whether a cell goes into the proliferation process. And it's translational control that the PI3 kinase AKT mTOR pathway is going to be involved in. So basically, normally, if you just activated the transcriptional control, if you just made all these new mRNAs for the proteins involved in making the cell divide, these pro-proliferation proteins, then it wouldn't actually have any effect, because normally there is a blockade on these pro-proliferation mRNAs being translated. Normally they're not allowed to be translated. Okay, the PI3 kinase AKT mTOR pathway is the pathway which says, okay, let them go through, let them be translated, and therefore these two pathways, the RAS-RAF-MEK-ERK pathway and the PI3 kinase AKT mTOR pathway, they control the two major controls of epigenetics together, and they allow the initiation of the growth and then the proliferation pathway. Okay, so it's important to understand that these work together. Okay, right. So that's an overview of what this pathway does, and hopefully you'll now appreciate how important it's going to be for cancer cells that want to over-divide to have some overactivation of this pathway along with a bit of overactivation of this pathway. Okay, so that you can then divide. Okay, if you don't have overactivation of this pathway, then even if you have overactivation of this pathway, you can produce as much mRNA as you like, but if you're blocking it from being translated, then it's not actually going to cause the cell to grow and then divide. Okay, so this pathway is extremely important in controlling the initiation of cellular division, and therefore we can understand that it would be a key thing that's going to have mutations found in it in cancer cells. And indeed it is. We find a huge number of cancer cells with mutations in components of this pathway, as we'll discuss later. Okay, so in the next video what we'll do is now discuss this pathway in detail. So we'll start by discussing phosphoinositide 3 kinase enzymes, PI3 kinase enzymes. Okay, then we'll discuss all of the different ways that PI3 kinase enzymes can be activated, because it is not just by receptor tyrosine kinases. And then we'll move on to the next portions of the pathway.